marker activated.
Okay.
So there's no...
So yeah, when you're done, I'll flip mine on.
Yeah. No, it should be fine. Yeah, oh yeah.
Okay.
Absolutely.
So I'll stand with it.
I would suggest going to the podium.
There, right here?
Yeah,
Okay.
yeah, either way, wherever you're comfortable. Because the camera, there's two cameras, there's one there and there's one there. Oh, no, right there. So, that's the idea. So about a minute?
Minute. Minute? I'll start.
In about a minute.
Oh, okay. So tell me when I have to start.
Yeah, about a minute.
Okay.
Do you have a smooth flight?
Yeah, very nice.
Well, that's good.
Yeah, if I get us to get down, that's good.
Yeah, yeah, that's good.
Start? Oh. Good afternoon, everybody. I'm Nargis Jarin from Research Division at ARB. Thank you, everybody, for attending today's Chairman's Seminar. The series is being webcast. We'll hold the questions until the end. I would like to encourage our webcast viewers to send in their email questions during the broadcast for the QA portion of the seminar to coastalrm@calepa.ca.gov. It is my pleasure to introduce today's seminar, Dr. Mark Rydell. Dr. Rydell is an assistant professor of medicine and head of clinical immunology and allergy in the Department of Medicine at the UCLA David Given School of Medicine. He is a diplomat of the American Boards of Internal Medicine, Allergy and Immunology, and Clinical Pharmacology. His primary research interests include the development of novel therapeutics for the treatment of allergic and immunodeficient conditions and the investigation of compounds to reduce the inflammatory effects of air pollution. The study, which Dr. Rydell will be presenting today, was funded by ARB, and this investigates the short-term health effects in susceptible volunteers from exposure to concentrated ambient particles. The title of the study is Differences in Inflammatory Responses to Exposures from Concentrated Ambient Particles in Susceptible Volunteers. Please welcome Dr. Rydell for his presentation. Dr. Rydell.
Well, thank you very much for that kind introduction. I'm very pleased to be here today to have the chance to speak to you about our work that was sponsored by the ARB, and also to be speaking to really such a knowledgeable audience. And certainly many of you know this field as well or better than I, and have done a lot of the work that I'm going to speak about today. I also want to say that by way of background, I am a clinician. I split my time between seeing patients and doing clinical research. And so my personal interest in this project and other projects that we've done is really trying to translate our findings to ways to help people in the clinic. Certainly living in Los Angeles, as you well know, we have an enormous burden caused by air pollution, and on a daily basis in the clinic I see people who are affected by this in terms of their asthma, their allergies, and their respiratory conditions. So that's my personal interest in this. But without further ado, these are the few areas that I hope to cover in the next hour or so. I will provide a little bit of background for the adverse respiratory effects of air pollution, and I know that with this audience you know this very, very well, but I do want to provide some context for the study that we performed in Los Angeles. I'll briefly touch on the concepts of the inflammatory effects of air pollution, and with some focus on individual susceptibility, because this is really what our study was designed to look at, is can we pick out who's susceptible to the adverse respiratory effects or health effects of particulate air pollution. And I think, of course, that's a very relevant piece of information to translate to clinical medicine. If we know who to target, we can actually use therapies that may be useful for subsets of the population. I'll then get into the actual study design and the results from the exposure study that we performed, sponsored by the ARB, and then finish up with some implications of our findings for future research. So, you all work sort of daily with air pollution, and I always like to start each talk with this phrase. We like to think of air pollution as a very modern problem, but in fact there are descriptions back to the Roman Empire of pestilential vapors and soot. So this is not a new problem. Our understanding of it has certainly increased, we hope, and we've gotten a little more refined in how we define air pollution, but certainly it's been around for quite some time. And as you well know, there are various flavors of air pollution, but we're going to focus mostly on particulates today because that's the study that we performed was looking at fine particulate air pollution. And this doesn't, there we go. Didn't show it first. This is just a reminder of the particle sizes. Our study that I'll be speaking about really dealt with fine concentrated air particles, and those of course are the respirable particles that we have great concern about in the respiratory tract. Certainly ultrafines are an area of great concern as well, that we did not focus on that particular specific type of particulate air pollution. A reminder of the clinical significance, and again, I will keep coming back to this. It's not to bore you, but this is why I do what I do, and I think why you do what you do, is that the air pollution has harmful clinical effects on human health. So epidemiology is wonderful. It's been able to show us that air pollution is convincingly linked to overall mortality from cardiorespiratory causes. There's no substitute for epidemiological studies because they have enormous power to look at large groups of people over time. There have also been a number of studies showing particularly air pollution is associated with pretty much any aspect of asthma that you want to consider: prevalence, severity, morbidity, medication use, and certainly hospitalization and mortality. The other point of interest from my standpoint as an allergist immunologist is now there's quite a bit of data showing that air pollution is linked to allergic sensitization. So people who live in highly polluted areas are more likely to produce IgE. They're more likely to become sensitized to environmental allergens. And we believe, although we haven't proven this, we believe this is one of the reasons that allergic diseases have become more common in the last couple of decades. The other thing I'll just remind you of is that there's no sort of threshold safe dose of air pollution. I mean, you certainly are aware of this, but this does appear to be sort of a dose-response effect that's linear, and so we have to take that into account when we look at these particular studies. So the questions that I think are important to address when we talk about air pollution and respiratory diseases: is there a link? I think I won't spend time belaboring that. There's a plethora of data showing the link between pollution and airway disease. But the second point would be the mechanisms. And again, you're likely familiar with this, but just to put our work into context, there have been a number of human-controlled exposure studies with regard to pollution. I'm just going to touch on a couple with diesel particles. Certainly diesel particles have been probably the most well-studied particulate air pollutant. And this is just a study out of Scandinavia demonstrating what has really been seen in human subjects with exposure to diesel particles. You see increases in inflammatory cells of the airways, increases in histamine and inflammatory cytokines and adhesion molecules, and increased airway resistance. What's also been noted, though, is that asthmatics seem to respond in a somewhat different manner, at least in some studies. And they have increased hyperresponsiveness. They still have increases in resistance and some inflammatory mediators, but not always an increase in airway cellular inflammation. And this increase in IL-10 is interesting from an immunoregulatory standpoint. And then lastly, I'll say that there have been human studies showing that the pathway that likely leads to inflammation, and some of you in this room have worked on this particular pathway, but that the activation of this redox-sensitive transcription factors is likely to lead to a pro-inflammatory and pro-allergic response. And so as an allergist, a clinical allergist, we have seen clearly demonstrated in a number of both animal and human experiments these four important effects of diesel now, speaking specifically about diesel, on allergy and inflammation in the airway. And that includes immediate effects. So exposure, within minutes of exposure to diesel particles, you see increases in histamine and increased symptomatology. You see a short-term response which occurs a few hours after the exposure, with chemokine and cytokines increased and increased cellular inflammation. You then see this intermediate-term response that Several days after the exposure, you'll get enhancement of total and allergen-specific IgE. And again, that speaks to the adjuvant effect of air pollutants, or in this case, diesel, on the allergic response. And finally, a long-term response, and this is enhancement of primary allergic sensitization. So that's to say, if you expose a genetically predisposed person to an allergen, a certain percentage will become allergic to it or make IgE. If you take that same person, that same allergen, and put along with the exposure diesel particles, you will significantly increase the percentage of patients that become allergic and make IgE against that allergen. So this is a strong adjuvant effect that leads to an increased prevalence of allergen-specific IgE in the predisposed atopic population. And this is just a schematic. It's a little bit complicated, but I think I sort of just wanted to highlight, because this does apply to particulates as we understand it currently. You certainly have, in this person here, this individual that's not allergic and doesn't really have the genetics for allergy, you still see direct effects of these particulate pollutants, and we've seen that in a number of human studies. Increased inflammatory mediators, increased airway resistance. However, if you now take a different individual who is predisposed genetically to make IgE or to become sensitized from an allergic standpoint, and you now expose them to allergen along with particulate pollution, you not only have the direct effects, but you increase the risk of primary allergic sensitization. So this is the adjuvant effect that leads to the development of IgE to a particular allergen in the setting of air pollution exposure. This next individual is someone who already makes IgE to ragweed, for instance, or cat, but they're largely asymptomatic. And through a variety of studies, we now know that if you take that allergen and expose a sensitized person along with a pollutant, such as particulate matter, you will increase the allergic inflammation or the histamine and other cytokine release that's associated with allergen exposure. So this essentially may take an asymptomatic allergic person to someone who has clinical symptoms of allergy, whether that's hay fever, sinusitis, or asthma. And then finally, in a person who has asthma, as you well know, we know that air pollution and particulates specifically do augment symptoms and do augment allergic inflammation with allergen exposure. So we have these 4 steps at which it's fairly clear from both animal and human studies at which particulates play an important role in allergic inflammation and in respiratory health. Importantly, the asterisk in each of these individuals indicates host susceptibility factors. And some of these we know fairly well, and many of these I don't think we've identified yet. But these include things like age, genetic polymorphisms, in a variety of points that may counteract inflammation. And this was part of our goal with the study, was try to identify susceptibility factors that may lead to increased inflammation with particulate exposure. Now, I won't go into great detail about the biology or the mechanisms by which particulates cause inflammation, but I do want to say a couple of words just to highlight the role of oxidative stress. As you well know, particulate matter contains a soup of pro-inflammatory or pro-oxidative chemicals, and these oxidative chemicals cause oxidative stress through the generation of reactive oxygen species. Now, we know that individuals or the human body has various cytoprotective mechanisms, but in the setting of increased oxidant stress, these protective mechanisms are overwhelmed, and you get resultant inflammation. An enormous amount of work has gone into this oxidative stress model, or hierarchical oxidative stress model. This is clearly an oversimplification of the mechanisms, but I think it's a very nice summary of how we understand oxidative stress and how it causes inflammation in the airway. If you see here, you start with the normal individual, and as you increase their exposure to oxidant chemicals in the form of air pollution, you ramp up the level of oxidative stress and you begin to deplete glutathione. Now, the body has a variety of antioxidant defenses, including the NRF2 pathway and the antioxidant response element. However, in the setting of increased oxidative stress, you get increased inflammation, and eventually will lead to activation of transcription factors that lead to cytokines which are pro-inflammatory and, in many cases, pro-allergic, including IL-4, IL-5, IL-13. ultimately, if you continue down this pathway of oxidative stress, you will get cell death and toxicity. But where we're really interested in studying is these two phases where we have an antioxidant response that's protective against oxidative stress, and better understanding how this inflammation occurs so that we might be able to prevent it. So the next question then is, how do we decide who's susceptible? And just merely as an anecdote, I certainly see every day in the clinic patients who are very susceptible to air pollution and those who don't seem bothered by it at all. And that holds true for people with asthma and without asthma. So this is part of the genesis of the question that we have, is how can we identify and test for susceptibility to air pollution? I show you here some data from Frank Gilliland and David Diaz-Sanchez that was published in The Lancet a few years ago. And this shows us that, in fact, if you take allergen, in this case it's ragweed allergen, R-W-I-G-E on the side here. These are subjects that are allergic to ragweed. If you expose them to ragweed in the nose, you will see some IgE production. However, if you expose them to ragweed along with diesel particles at the same time, you can see, as I've mentioned, this enormous boost in IgE production, indicating an adjuvant or boosting factor of the particulate pollutant. However, what you'll also notice is that not everybody reacts the same. Some people have an incredibly robust response, up to 700, from basically 100 to 700, and others basically stay the same with only a marginal increase in their IgE production. So this is precisely the question we're trying to get at, is how do we identify who's a robust responder? We do know that, or we believe that children and the elderly are at risk for respiratory conditions due to air pollution exposure, probably in children because they lack a full development of the protective pathways against oxidative stress, and probably in the elderly because, again, they have some lack of defenses and also have a number of comorbid conditions that may complicate their cardiorespiratory status. But interestingly, it's not always the case that asthma severity predicts inflammation with air pollutant exposure. This is a little bit outdated study now from 1998, but it's very interesting in that this was a group that looked at asthmatics, classified them by severity, symptoms, and medication use, followed them over a period of time, and basically tracked to see how badly they flared or if they worsened with air pollution exposure. And what they found is that you could not predict who was going to have an exacerbation with air pollutant exposure simply based on how bad their asthma was. There were certainly mild asthmatics who had a great deal of difficulty with poor air quality. So to sum it up, there's pretty clearly inter-individual variability in inflammatory responses, at least when we study diesel. And this just shows that this variability is fairly reproducible in the individual. So on the x-axis here, you'll see the first visit and the first exposure to allergen plus diesel. And on the y-axis, you see the second visit where the same procedure was repeated. And we have a pretty nice correlation here in terms of the reproducibility of this response, suggesting that this is really an intrinsic host factor. This is some additional data from David Diaz-Sanchez's work at UCLA, showing that if you look at any variety of inflammatory markers with regard to diesel exposure, you have a group that very clearly responds in a robust fashion for pretty much any marker you want to consider. And this is what we would define as a susceptible phenotype. You also have a group that seems fairly well protected down here at the bottom. And then, of course, there's a lot of noise. There's a lot of variability for other subjects. But it's this group here, this susceptible phenotype, and perhaps this protected group, that we're most interested in studying to see if we can tease out factors that might indicate whether a person is really at risk for health effects from air pollution. And then I'll jump to this to show you that same study that I indicated earlier, but now filling in a variable, which is whether this person had a functional GSTM gene, and this is GSTM1. And it stratifies very nicely, showing that if you make this GSTM1, this phase 2 enzyme that we think plays a role in metabolizing and neutralizing oxidative stress, that you don't have a particularly robust response to the diesel exposure. those that lack this GSTM1 genotype or are null for this polymorphism have the most robust responses. And so if you look at other outcomes besides IgE, this continues to hold true. If you look at histamine, if you look at total IgE, again, those that lack this enzyme have much more robust inflammation compared to those who make the protective protein. And so this led us to really look at the glutathione-s-transferases as a potential area where people vary with regard to their response to particulate air pollution. As a group, these are phase 2 metabolizing enzymes. We think they're important for protection against xenobiotics. And they do two important things that we know of so far. They detoxify these chemicals or metabolize the chemicals that are found on particulate pollutants. And they also neutralize or metabolize reactive oxygen species. We've gone on in our laboratory to replicate this using some in vitro methods. This is simply looking at human bronchial epithelial cells, and you can select cells that have both expression and non-expression of this GSTM1 loci. If you take those cells and you expose them in vitro to diesel, you can then measure the cytokines and really quantify their responses to diesel exposure. And this is just some unpublished data from our laboratory showing that if you look at diesel exposure and look at IL-8 production by these cells, it doesn't take statistics to show that this is a very robust response in the negative GSTM1 cells, and really some, but not much difference in the cells that make GSTM1. And you can do the same thing for other inflammatory cytokines, including IL-1 beta, where we again see GSTM1 negative cells having a very robust response. We've replicated this actually using siRNA to knock out expression of GSTM1 in other cells, and we get basically the same results. So I show you this to show you that this is data that has suggested that this particular loci or polymorphism in GSTM1 may be a candidate for susceptibility and identifying those who are at risk for air pollutant inflammatory effects. The last little background piece that I want to mention is, again, something I probably don't need to mention, but for those who may not be familiar, we used concentrated air particles in the study I'm going to tell you about in a moment. Of course, the benefit of this is that these are real-world particles. This is not concentrating diesel and using it in a fashion that might only be seen in certain occupations. This is real-world particles drawn from the air and used for exposures. And as you likely know, these are complex mixtures. They include dust from soil, they include sea salt, it includes combustion particles from engines, from tires. It's a soup. It's a mix of all kinds of particles. Time of year that they're gathered. And we know that these particles, from a number of analyses in the past, including those at our site in Los Angeles, we know that they contain elements that I've listed here, and also reactive surface compounds, including polyaromatic hydrocarbons. And that's likely important because it's not entirely clear from the literature, but much of the inflammatory responses seem to be associated with these either metals or the polyaromatic hydrocarbons. So in terms of designing our study, we looked, of course, at studies that have previously been done. I list here some, but not all, of the human exposure studies that have been done for concentrated air particles. And actually, most of these were done, at least in part, by the site in Los Angeles that we worked with. But you can see here the caps have been used for both healthy and asthmatic volunteers, including ultrafine particles, looking at both respiratory and heart rate variability outcomes with regard to particles. And also, I just want to highlight that you have fine, coarse, and ultrafine particles. So again, there are many variables that can be looked at here with regard to particle size, particle content, the population that's looked at. So I'll now move into our study design and our study results. By way of background, let me say that this study was conceived, written, and proposed by David Diaz-Sanchez, who's now at the EPA. David departed UCLA sort of in the middle of this project, and so I was pleased to have the opportunity to work on it, or sort of take over the heavy lifting, if you will. But I do want to make sure that he gets the credit for really the concept and a lot of the details that were put into this particular project. Our interest, as I've mentioned, was to see if we could identify susceptibility factors that would predict inflammatory responses to concentrated ambient particles. And so we worked with Bill Lynn and Ken Clark, who are at Los Amigos Ranchos Research and Education Institute in Downey, which has a vast experience doing human exposures with several types of pollution, including diesel and ozone and concentrated air particles. And they have a great deal of expertise in this area. We also worked with statisticians from USC who have a lot of experience analyzing this very complex data that's produced. So our specific objective was to test the hypothesis that individuals with certain susceptibility factors have heightened inflammatory and airway responses to exposure to concentrated particles. And the susceptibility factors that we decided to study were underlying asthma and then this absence of GSTM1 production. So we genotyped patients and determined whether they had GSTM1 present or GSTM1 null polymorphism. And this, of course, is based on the data I just showed you from the upper airway, looking at the susceptibility in the upper airway, and also a number of epidemiologic studies that have showed that GSTM1 null may be a risk factor for asthmatics living in polluted areas. The design was a single-blind, randomized crossover study comparing filtered air exposures to concentrated ambient fine particle exposures. These exposures were 2 hours in duration, and the subjects actually exercised on a stationary bicycle for about 15 minutes of every half hour. This was, of course, to increase their ventilation. The target concentration for the air particles was 200 micrograms per meter cubed, and we enrolled 3 distinct groups. And the idea here was to try to have a low, moderate, and high-risk group of patients to study. So the high-risk group would have been these 10 GSTM1 negative asthmatics. So they have, we think, 2 susceptibility factors. They have asthma, and they lack this protective GSTM1 enzyme. The moderate risk group would be GSTM1 positive asthmatics who have underlying respiratory disease but make the protective GSTM1 enzyme. And finally, the low risk would be healthy subjects who also have protective GSTM1 expression. As I'll show you, we measured quite a number of inflammatory and airway responses, as well as some systemic inflammatory markers, and also did Holter monitoring to see if we could detect any cardiovascular changes with these particulate exposures. This is just a brief outline of the study design. These patients came for, or subjects came for screening visits. Then 2 weeks later, they underwent an exposure, and that exposure could have been ambient particles or filtered air. Those were randomized for each subject so that it wasn't predictable which one happened first. They then had a follow-up visit, and at least 2 weeks later underwent the other exposure, whether that be filtered air or concentrated air particles, which was also followed up by a visit for data collection. And this is a busy slide. I show it to tell you that we tried to be comprehensive in looking for outcomes, important outcomes that may indicate inflammation in these subjects. So at baseline or pre-exposure, we did Holter monitoring and EKGs, vital signs, blood draws. We did exhaled nitric oxide. We also did spirometry and urine collection. And I should say at screening, these patients underwent a baseline sputum collection. We did not want to do that at the baseline pre-exposure because of the possibility that that would affect the day 2 sputum collection. But we did have baseline sputum to collect for comparison for the post-exposure samples. We also wanted to look in the upper airway to see if we could detect changes in both the upper and the lower airway. That was the purpose of the nasal lavage that was collected. And finally, there was a symptom score sheet that each subject filled out. This measured respiratory, cardiovascular, and some what I would call irritant symptoms that patients might feel. So that would include burning eyes, burning nose, those sorts of things that might not be captured by any of our other measurements. So after the exposures, the subjects underwent a variety of post-exposure collections in order to compare. That included a methacholine challenge with spirometry. And then finally they returned on day 2 after the exposures for this list of additional collection of biomarkers in order to do the comparisons for pre and post exposure. And so again, just to show you the biological endpoints we looked at, we were really trying to capture as much as we could, including vital signs, bronchial reactivity, a number of respiratory outcomes, including functional measures of spirometry, exhaled gases, the sputum cytokines and antibodies, the nasal lavage cytokines and antibodies. We did measure some systemic markers, including C-reactive protein and a number of factors in the coagulation cascade, and then urine for 8-isoprostanes, and then, as I mentioned, the cardiovascular data to look at heart rate variability changes. In terms of the specific exposures, these were done in a whole body chamber. These were, as I'll show you in a moment, these were largely fine particles that were used for the exposure, and the measurements of the exposure were monitored both by real-time nephelometry, but also filters were taken to measure mass and particle size. As you're probably more familiar with than I, the mechanism for this was a concentrator that is actually very closely resembles that used by the EPA. And the particle concentrations were controlled by diluting output of the ambient fine concentrator with amounts of filtered air. The actual setup in Los Angeles or in Downey, California, draws the air from the roof, and it's about 4 meters above the roof, and we're able to concentrate these particles up to 9 times. The actual physical area where this is done, and there's, as I mentioned, a vast experience with these types of exposures, but this is an area in southern LA County that has a fairly high background pollution level due to very busy surface streets. There's also the 710 freeway runs very near there, which has a lot of diesel trucks on it. And the Long Beach port is just about 10 miles south, and as you may be well, is a real trouble spot for air pollution in the Los Angeles area. So there's been a great deal of previous work. I've mentioned some of the references here on the slide, but characterizing the neighborhood's pollution and the air particle exposure atmospheres. We know that in the past, organic carbons, elemental carbon, nitrates, sulfates are all major constituents of these particles. And we did measure particle size distribution in a variety of ways, which I'll show you in a second. The filtered air exposures were obviously the control arm, and these were done really in the exact same manner, using the same chamber, except that the ambient air was filtered using a HEPA filtration system. We also did monitor the carbon monoxide, NIOX, and sulfur dioxide and ozone levels, so that we could keep track of those with the various exposures, though previous testing has shown little difference between the ambient and in-chamber measurements of these gases. So that's a bit about the methods, and I want to show you basically the results of what we found. This is the population that we enrolled. As I mentioned, there were 10 in each of these groups, and you can see that they were largely female and largely in the 30s and 40s. There was a slightly older age group in the asthmatics that were GSTM1 positive, but overall no other major differences. And the ethnicity really reflects the local population, and that we had a very large Hispanic group of subjects. These are the environmental measures that were taken during both the filtered air and the concentrated farm particle exposures. So you can see based on the filter measurements that we actually came very close to our target in general at a mean of 187 micrograms per meter cubed, the target being 200. The nephelometer measurements didn't exactly measure up. They actually were a bit higher with comparison to the filter measurements. The rest of the variables were actually quite close. The only one I do want to draw your attention to is the the nitrogen dioxide levels, those actually were slightly higher in the particle exposures. And the explanation for that is that there were days when we had scheduled particle exposures where the ambient particle levels were too low in order to do a good exposure. And so that exposure had to be postponed or rescheduled. And so you do have days that were sort of low pollution days, if you will, where filtered air exposures may have been done where particle exposures had to be postponed or canceled. And because particles tend to track with nitrogen dioxide in our area at least, that's an explanation for why the particle exposures seem to have a slightly higher NO2 level compared to the free filtered air exposures. Otherwise, the other meteorologic measurements and gas measurements were basically quite similar. This shows the particle mass versus size range is determined by the impactor sampling. And you can see that the vast majority of these particles did fall into the fine range. There's a very small amount of ultrafines and some amount of more coarse particles, but the vast majority did fall in the fine particle range. And this compares the particle exposures with the free air exposures, and you can see that obviously there are much greater particle exposure during those sessions compared to the filtered air. There is some coarse particle measurement out here for free air, and we think that represents particles from subjects' clothing and the activities that were done in the chamber. This is a very busy slide, but it's actually very important. And so I just want to, for a moment, run you through it. This is, of course, looking at the chemical analyses of the particulate samples of our exposures. And what I want to point out, that now, certainly if you compare the particle exposures to the free air exposures, for most things that we think are important, there was significantly higher levels of both elemental, organic carbon, as well as a number of important metals. So certainly you have higher exposures with the particles than the filtered air. However, one concern that's come out of our data analysis is that these levels of the metals in particular are significantly lower than we've seen with our other particle exposures at this site. The ECOC looks about the same as what we've seen previously. But if you look at these, and you have to actually do a little math to get the nanograms per liter to the micrograms per meter cubed of air. But these equate to basically, even for the ones that are higher, so calcium, iron, aluminum, potassium, these equate to really nanogram levels per meter cubed, as opposed to microgram levels that we've seen in previous exposures at our site with concentrated air particles. So this is potentially a real issue in that qualitatively our particles seem somewhat different from what we've had with previous studies at our Los Angeles site. I'll shift now to some of the subject characteristics, and this shows you pre-exposure physiologic and symptom measurements by group. And again, for the most part, we don't see dramatic differences between the three groups. I will point out that symptom scores seem to be, at baseline, seem to be slightly higher in particularly this group with asthma and GSTM1 expression. I'm not sure how to explain that, but that's the data. Secondly, I do want to point out that the FEV1 to FVC ratio is lower in the asthmatic groups, as you might expect. However, it's not profoundly low. And so I didn't mention this specifically, but these were mild to moderate asthmatics. They were required to have an FEV1 to FVC ratio of greater than 70% because we did not want to put more severe asthmatics at risk for health problems from these exposures. So I think that the mild state of asthma in these subjects is reflected in the fact that they do not have profoundly low FEV1 to FVC ratio. That's important to consider. And the other thing I will point out is that the exhaled nitric oxide is higher in the asthmatic groups compared to the healthy groups, which is as expected. We know that FeNO is elevated in patients with asthma. So again, that's somewhat reassuring in that we did capture patients that had existing airway inflammation, at least if you base it on FeNO measurements. This is a mixed model analysis of the physiology and symptom data. And the real point I want to make here is that we did not see dramatic differences or significant differences in the physiology and symptom data that could be attributable to the particle exposure for most any of the measurements we looked at. You will see here that there are some significant differences for symptom scores during and after the exposures, and those seem to fall to the susceptible group, if you will, the GSTM1 negative asthmatics. However, these were the same if the patient underwent a free air exposure or a particle exposure. So it really seems to be an effect of the exposure protocol itself rather than the particles having that specific effect. Same can be said for the drop or the change in systolic blood pressure, which was significantly different in the GSTM negative asthmatics, but didn't seem specific to particles, but rather to the exposure protocol. The only area we did see a difference, which we could attribute statistically to particle exposure, was this change in exhaled nitric oxide. And I think I'll talk about this more in a minute. This may be interesting. It may be something that we need to look at more closely. But this was an area that across the groups we saw a difference with particle versus filtered air exposures. I'll just show you, I like pictures, easier for me to graph sometimes, so I'll show you a couple pictures of the data. This is looking at the change in systolic blood pressure pre and post exposure. And again, as you can see, there are some differences between groups. You can see that the systolic blood pressure dropped less in this GSTM null asthmatic group. But if you look at free air versus particles, there's basically no difference between that specific exposure variable. And so, again, I don't think we can attribute that to a particle effect. This is the exhaled NO data. And what I will show you here is that if you look at the entire group, these are all the subjects together, you can see that this change in NO was significantly different with the particles compared to free air. So you see a drop in NO with free air and less of a drop with particles. Now the drop is not necessarily surprising. These patients were exercising, and it's actually been shown in a number of studies that exercise will decrease exhaled NO levels. However, if you start to look at the actual asthmatic groups, you can see that a number of asthmatics, based on the spread here, some asthmatics did show an increase in their exhaled NO with the particle exposure, where pretty much across the board you saw this consistent drop with filtered air exposure. And this increase, or in one case, the mean was less decreased here, was basically consistent for the asthmatic groups. The healthy groups didn't show that significant difference. So I'm not exactly sure how to interpret this. I think it is a finding that seems to be specific both to the asthmatic groups and to the particle exposures. Unfortunately, as you'll see, we don't have a lot of consistent other inflammatory data to sort of go along with this finding of exhaled NO. The response measures, we tried to correlate these with various other factors that might be important, including concentration of particles, NO2 exposure, and chamber temperature, and we really didn't see any strong correlations. The only one that's significant was for symptom score and nitrogen dioxide, but you can see that actually goes in the, quote, wrong direction, meaning that symptoms got lower as NO2 went up. So I think that's probably just a random finding rather than a mechanistic finding. Now we'll look at some of the sputum or respiratory data. This is looking at neutrophil percentages along with monocytes and lymphocytes in the different groups. What you can see is that there was a statistical difference with this GSTM positive asthmatic group. They had more neutrophils at baseline and also after the exposures. However, the GSTM negative asthmatic group did not have that difference compared to the healthy controls. You can also see a strong trend, and I believe the p-value was less than 0.1 but not less than 0.05 for neutrophil increase with particle exposure. So we had a strong trend towards increased neutrophilia in the induced sputum across all groups, suggesting that there very well may be a biological effect, but not reaching statistical significance and not different between the susceptibility groups with this exposure. This is looking at IL-4 levels in the induced sputum, an important cytokine for asthma, both from a pro-inflammatory and pro-allergic standpoint. And this just points out that at baseline, the asthmatic patients had significantly higher sputum IL-4 levels compared to the healthy subjects. Not surprising, that's what we would expect to see. Unfortunately, we did not see a significant increase with particle exposure compared to free air exposure, though you did continue to see this significant difference between asthmatics and healthy subjects for sputum IL-4 levels. This slide shows other biomarkers in the sputum that we thought would be important to indicate a pro-inflammatory response from the particle exposures. Again, this is looking at all subjects. And the bad news here is that we did not see a lot of statistical differences in the biomarkers when we compared free air to particle exposures. We did see trends if you look at, for instance, interferon gamma, GM-CSF, tumor necrosis factor alpha. You can see that there's certainly more subjects that had measurable levels of these inflammatory markers, but we didn't reach statistical significance for these particular markers. So overall, the sputum data was somewhat disappointing in that we didn't see large differences from particle to filtered air exposure, nor from susceptibility groups when comparing them. I mentioned earlier that we were interested in the upper airway as well as the lower airway. We did a number of measurements on nasal lavage fluid to see if there were either consistencies or differences from the upper to lower airway. And for the most part, the nasal lavage fluid findings were negative, meaning that we didn't find any significant differences. The one exception to that was nasal lavage IgG4 levels. And this shows, this is a bit busy, but I'll focus you on, this is the overall group numbers, this dark line with the black diamonds. This shows that with particle exposure, you saw a significant increase in nasal IgG4 expression. And with free air exposures, we saw basically a decrease. And so that holds true for all of the susceptibility groups in that particles induced an increase in nasal IgG4, while free air either tended to hold stable or decrease. So that was a difference that we saw from free air to concentrated air particle exposures. We then took a look at the different sputum outcomes to see if we could associate those changes that we saw with any of the particular particle elements or particle characteristics. And for the most part, we did not see any dramatic associations. You can see that some of the elements do track together in terms of the levels. They were statistically associated with one another. But in terms of the biological outcomes, there's very little that can be said with regard to what might be causing these particular biological observations. There was some indication that, for instance, IgA and IgG4 might be associated with either mass or chromium content, but other than that, these were largely negative or non-significant associations. And I'll finish with regard to the biological outcomes with heart rate variability. Now, I'll just be honest with you, I am not a cardiologist, and I find heart rate variability to be quite complicated with regard to both the number of things you can measure with Holter monitors and also the statistics that need to be done to look at changes. I've listed here on the left just a number of things, a number of outcomes that were analyzed to see if we could detect any heart rate variability differences between groups or between exposures. And on the right, I've shown you a graph of really the most significant difference that we found, showing that across all groups now that there seemed to be an increase in T-wave complexity post-free air compared to concentrated air particle exposures. Now, unfortunately, this is in, again, the wrong direction. Increases in T-wave complexity are believed by some to be a risk factor for arrhythmias and may contribute to adverse cardiovascular outcomes. So one way to interpret this is that the free air actually, or the filtered air actually had more detrimental outcome on this particular measurement compared to the particles, which intuitively doesn't make any sense to us. So these are the overall conclusions after a lot of hard work and effort by large groups of people. We were not able to demonstrate clear, robust differences between the exposure responses of these normal or low-risk subjects compared to the susceptible subjects. There is the exception of the increase in exhaled NO for all groups, which was a significant biological finding. However, other than that, the particles exposure as performed in our study doesn't appear to produce a robust inflammatory, either respiratory or systemic response in human subjects, at least using the biomarkers that we measured. And furthermore, we did not find that subjects with asthma or this missing GSTM1 expression were more susceptible to the inflammatory effects of these concentrated air particle exposures. There were significant baseline differences for exhaled NO for the asthmatic and healthy subjects, so I'm reasonably confident that we captured asthmatics versus healthy subjects. We did have a trend, but non-significant difference in the difference in FEV1 for the asthmatics versus the healthy subjects. But as I highlighted, there wasn't a dramatic difference. It was actually statistically non-significant, really, I think, pointing to the mild asthmatics that we used in this particular study. And ultimately, there were a few differences in responses with the exposures that we could attribute to the particles themselves or between the susceptibility groups. Just a word on this, the one finding of exhaled NO. This was increased with the particle exposure versus free air. However, it didn't differ based on the GSTM1 status or the asthma status, though the increase was greater in the asthmatic versus healthy groups. This has been reported in observational studies looking at air pollution levels and tracking exhaled NO in the population. So on a very large scale, observationally, we have seen this before. However, the previous concentrated air particle studies, controlled human exposures, have either not looked at this with fine particles, or in the case of other particle sizes, such as coarse or ultrafine particles, they've not seen these consistent increases in exhaled NO with particle exposure. So that's to say that I'm not certain what the significance of this finding is. It may be that we have stumbled on something that may be important, an important measurement to track responses in asthmatics or in healthy subjects to particulate air pollution, but this is not something that's been replicated or seen with other particle sizes and other controlled studies. In terms of the sputum biomarkers, we did see that one group of asthmatics had significantly higher levels of both neutrophil counts, sputum IgA levels, and lower levels of monocytes. This was really a difference between the GSTM positive and the GSTM negative asthmatics, and I don't have a good explanation for that. We know that asthmatics have a variable phenotype, if you will, based on the amount of neutrophils and other markers in the lungs. It's not been described that this is linked to GSTM1 status in the past, so perhaps this is something that we're newly describing, but I just don't know enough in this small group to say whether that's the case or not. We did not see that these biomarkers were affected by the exposure conditions. And the reason I think this might be of interest is that we did see significant group differences for sputum IgA with the particles versus the filtered air. And this was really an increase with healthy subjects. GSTM positive asthmatics basically stayed the same, and GSTM negative asthmatics showed a decrease with the particle exposure. Now again, this is going a bit into uncharted territory, but we did see an overall group variation with the particle exposure that approached statistical significance when looking at sputum IgA levels. And this wasn't something that I would have predicted a priori, but it is a bit intriguing in that sputum IgA is now increasingly being recognized as an anti-inflammatory antibody, both in the systemic circulation and in the mucosal surfaces. I've listed a couple of references here at the bottom that have looked into this. So one could speculate that because the healthy subjects had the highest increase in this, and the GSTM negative asthmatic subjects really had a decrease with particle exposure, that there might be either some deficit in IgA production at mucosal surfaces, or potentially that this is some way linked to GSTM1 status. This is completely speculative. I don't have any other evidence to suggest that link, but it is an observation that we saw in our study, and it would be interesting to perhaps investigate this particular association on a larger scale. With regard to sputum IL-4, the asthmatic groups did have higher baseline levels compared to the healthy controls, and those levels continued to be higher with the exposures. However, we saw overall decreases in the sputum IL-4 levels with any type of chamber exposure. And this is really in contrast with what we've understood about particle exposure, particularly with diesel, where you see that exposure to diesel causes increase in IL-4 production from T cells. I purposely did not show you a lot of the blood and urine biomarker data because we did not see any blood or urine biomarkers that showed exposure differences that were either different between groups or attributable to the particle exposures. Again, this contrasts with some previous human studies which have shown that you could measure a few serologic biomarker differences in either fibrinogen, D-dimer, or IL-8, with the caveat that some of these effects have been observed with ultrafines, and of course we studied fine particles in our particular exposures. And then finally, the heart rate variability, just to summarize, we know this is an important cardiovascular outcome, and that it may be a prognostic marker for cardiac arrhythmia. But the changes that we saw in our study that were attributable to concentrated air particles included a mild decrease in heart rate and decreased T-wave complexity and variability. And really, as I already mentioned, these are somewhat inconsistent with our previous reports and understanding of heart rate variability, and that other studies have shown increases in heart rate and increases in T-wave complexity with particle exposure. So our findings were actually opposite those that have been reported. So in terms, I'm going to just spend the last few minutes on limitations of our study and also, where does this point us for the future. One of the big questions, as with any exposure study, is did we have adequate exposures? And I think we clearly used a well-established protocol, a well-established facility that's done quite a number of these exposure studies in the past. The equipment was the same, the protocols were the same. And we did show, based on particle counts and concentrations, that we achieved the target concentration of 200 micrograms per meter cubed. But as I mentioned, there may be something qualitatively different about the particles that were used in this study. The ECOC results are consistent with previous exposures. It doesn't appear that those were far off from what we expected to get. But the PAH levels, which I didn't show you that data, but we did have a number of samples that showed basically undetectable levels for a certain number of PAHs. And across the board, the levels of these hydrocarbons were relatively low compared to other studies that have been done. So this could be an important contributing factor that would explain why we didn't see some of the inflammatory markers that we expected. The other area that could have been different was the transition metals and elements. And as I showed you, based on our analysis and calculations, we were really only achieving nanogram levels of these metals and elements compared to microgram exposures that have been seen with other studies, including those at our site that have been done in past years. There's no reason or no explanation that we have that would explain why these particles would be different qualitatively. We're not aware of any changes, substantial changes to the actual composition of the particles that are collected and used in these studies. But I do think it's important to recognize that as a very significant limitation that may explain some of our findings. The other limitations include the asthmatics, and as we've already discussed, these were mild to moderate asthmatics. They were not taking inhaled steroids, which by definition usually makes them mild, and they did not have a profoundly low FEV1. So I think that we have to take that into consideration. It may be that more severe asthmatics are much more susceptible to these effects, and that we may see greater differences if we studied that population. We also may be limited by the study power. As with any study, you have to look at that question. This study was designed to detect a 3% reduction in FEV1 using the sort of usual criteria of 0.8 of a one-tailed test with alpha of 0.05 and 10 in each group. As with many asthma studies, FEV1 was used as a clinically meaningful outcome, but it's certainly possible that may miss some of the more subtle changes that we may see in biomarkers, and those could have gone undetected. As I showed you, we did have some data that showed a trend towards significance, but did not reach statistical significance. We had many variables measured in relatively few subjects, and this is the difficulty of human studies. I sometimes wish that I studied mice, because mice are easy to do an experiment, and if it doesn't go well, you change it and you do it again the next week. And you're much better to control the variables with animal and in vitro studies. We don't have that luxury with people. There are a lot of biological variabilities. There are a lot of differences in environmental stresses that people have, emotional factors, all kinds of things that may affect some of these very sensitive markers that we're looking for in our study outcomes. So that is a limitation of any human experiment, particularly when we're studying a total of 30 subjects. And I do think we have to recognize that given the long list of outcomes that we measured here, there's always the possibility that you find some differences just by random chance, because if you look at enough variables, you will eventually find some that are statistically significant. So that's an important point to take into consideration. I've already mentioned biological heterogeneity, but I do want to highlight a couple of things. And this was brought up by some of the reviewers when we were submitting the final report. It's absolutely true that there are multiple variables that one should take into consideration when you're looking at humans. And so you can consider age, you can consider nutrition or dietary status. We tried to look at one genetic susceptibility factor, but clearly there are multiple genetic susceptibility factors, and I think there's probably a network of these susceptibility genes that come into play. We also have to consider ambient exposures that patients or that subjects have during their everyday life, and also their other possible comorbid diseases, although we tried to control for that and enroll just healthy subjects who simply had asthma. One point that was brought up by a reviewer, which I think is a very good one, was the obesity factor. And so this is a hot topic as to how obesity might actually contribute to inflammation or affect responses to air pollution inflammation. And we're not powered to look at this. It wasn't included in our original hypothesis. But just for fun, I went back and looked, and we had basically 7 or 8 overweight or obese subjects based on BMI in each group. So at least in this study, we didn't see dramatic differences based on obesity. But that's just an example of one factor that could come into play when we look at small groups of human subjects. And I already mentioned this, but the other cofactors, including genetics, that might modulate these inflammatory responses. Again, we looked at GSTM1 because of both epidemiological data and human data that suggested this was a very important modulator of particulate-induced inflammation. Unfortunately, we weren't able to demonstrate that as a strong susceptibility factor. And so that's something I think that we need to keep in mind as we go forward and design other studies looking at susceptibility to air pollution. So in terms of where this takes us, I think some of these are obvious. Anytime you can do larger scale experiments, you can have increased power. And of course, there are limitations when you talk about human studies. These are very expensive, they're very time consuming, and they do involve some risk to human subjects. So we have to balance all of those factors. But clearly, if we're able to study more human subjects, we may have more sensitivity to detect differences between groups. As I mentioned, there may be value to looking to other endpoints besides spirometry. That's sort of been the gold standard for asthma historically, but I think that there may be more sensitive ways to power these studies and look for specific biologic endpoints. With regard to the exposures, again, we may have run into a qualitative exposure issue with our particular study, but certainly differences in concentration or differences in duration of exposure may lead to different findings. And I think, as you're well aware, there has been efforts to try to standardize the exposures that are done at different centers so that we get comparable information. The severity of asthmatics I've mentioned, and I think that, again, there's ethical issues that need to be considered when you're enrolling asthmatics or any other susceptible individual. But clearly, the severity of disease may play a role in the outcomes that we see when we study these groups. The other genetic and cofactors, such as dietary intake, is fascinating literature on diet and nutrition in response to air pollution exposure. And while dietary supplementation doesn't seem to have been very effective so far in reducing the health effects of air pollution, looking at just dietary intake in general, meaning that eating fruits and vegetables does in some studies seem to be protective. And so I think that that may be a factor that we need to consider when we enroll subjects and need to correct for when looking at biological outcomes. And then I finally mentioned this inclusion of exhaled NO. Exhaled NO is an interesting measure that is still finding its place, I think, in the clinical management of asthma. I personally think it'd be interesting to continue including that as we study the effects of air pollution in both healthy and asthmatic subjects. I think we had maybe a somewhat interesting finding here, in that that was one of the few outcomes that seemed to be consistently different between the particle and filtered air exposures. I don't know the significance of that, but I think that's one of the reasons to continue to look at that question in various groups and various exposure conditions. So with that, I will finish. I do want to, again, give a great deal of credit to David, who really kicked this off and was the brains behind much of the work. I also want to make sure I thank the Bill Lynn and Ken Clark and their entire staff at Los Amigos Education Institute. They are the expert in the exposures, and they really did a lot of the heavy lifting when it came to the nitty-gritty exposure protocols. My laboratory technician at UCLA, Hema Shah, who did a great deal of laboratory work, and then Kiros at USC, who is really a world-class statistician when it comes to all the complexities of analyzing this data. So thank you very much for your attention, and I think we'll take some questions.
I have a question about the power. I realize that your power calculation, you use 1/2 p-value with 1/2. So when you present your result, is it everything based on 1/2 or 2/2?
I think 1/2.
So all your p-value is based on 1/2. So you do not care about what happened on the other side. You just strongly believe that only. I'm just wondering.
I'm not certain. I must admit that I'm not a statistician, and so I know that that's obviously how the study was powered on the primary outcome. But I have a feeling that for a lot of the biological outcomes that they did look at two-tailed, because we want to know what the other side.
They usually only strongly believe to go to one side. That's okay, but
so I think I misspoke. I mean, I think that was the primary powering of the study, but certainly for all the other sort of secondary and tertiary outcomes, I'm almost certain it would have been done two-tailed.
And then the n equal to 10, I do not use to run a sample calculation for mixed model, but I'm thinking about logistic regression. if I have a 3% change by using logistic regression to calculate a clinical trial, n equal to 10, I think it's very small. The sample size is not big enough. This is why you probably cannot detect any significant difference, even though your magnitude, the direction of magnitude do show something.
Yeah,
but the p-value do not tell you that.
And
This is what I'm
I
thinking.
think that's a very valid point.
Yeah.
I mean, I think one of our concerns all along, well, again, I wasn't involved in actually designing the study, but I think as we went along, and certainly once we looked at the data, we saw a lot of trends, but we didn't have significance. And I think, you know, that's always a problem with human studies because they're expensive. There's some, you know, issues with recruiting subjects. But I think you make a great point. Had we done N of 20 in each group, we may have seen something different. I mean, we may have certainly seen differences between the groups. So, yeah.
Hello? With a very small change, with an n equal to 10, I don't think you can detect the significant difference. This is what I'm thinking. It's just personal opinion. Probably that's the right number. I do not know.
No, and I'm with you. There was. I mean, I don't want to give the impression that there was a random number. There was a calculation done, and it was based on previous data that has looked at FEV1 change with exposures. And the reason FEV1 was chosen is because that's where the data was to power the study on. There wasn't enough data to use one of these other outcomes for the power calculation. So that's, I think that was the reasoning behind it. It was based on previous data that showed that we'd have enough power with 10 subjects to detect that change. But to your point, I think that's a concern. That's why I raised it as probably the first study limitation. This was a fairly small study.
Yeah, because the direction of the magnitude of the direction of the, what do you call it? The magnitude of the direction looked right. It's just not significant difference. So this is what I'm thinking.
Agreed.
One of my questions about it was that, in other words, your GST null mutant, you would expect them to actually have even more of an effect if you had higher levels of PAHs. It's very well known that the GST null individuals can't detoxify a benzopyrene ursodeoxyl epoxide, so they're going to be forming these benzopyrene addicts. Have you thought, that's the first question, have you thought about that, and particularly that you also have low levels of iron that you normally have, so you're not going to be having the Fenton reaction kind of things could be causing additional stress. Could you comment on the lack of the GST effect, given these background elements to what they might be if you had normal levels of those environmental exposures?
Yeah, well, I think, I mean, I agree. I think you made the point exactly. That's why I wanted to be clear about the sort of qualitative concerns we have with the particles, in that we didn't see the same levels of PAHs that we expected, and we didn't see the levels of the elements and the transition metals. And those are the things that one would expect to cause problems for GSTM null subjects. So I do think, you know, had the particles been qualitatively differently, again, that's another issue that may have made a dramatic difference.
The second question, if you're going to ask one more, is I think it's really important to be looking at the genetic susceptibility loci and everything, but in essence, the study design was assuming that GST mu1 was a single loci, kind of a major locus. In reality, we know that most these disease phenotypes are quantitative. There are multiple loci that are involved, and there's a number of genome-wide association studies that have really shown many different loci that are involved in those. And I'm just wondering, what would happen if you had a different study design, let's say in the future, whether you took, based on former genome-wide association studies, the individuals that would show the most susceptible genotype at multiple loci, go through a screen of population and now test those, not whether they were asthmatic or not, but rather their genotype. Because I think it's going to be really important in the long run to be able to include these genetic susceptibility sort of loci in the kind of risk assessments that we do as well.
Excellent point. I actually had this slide in there and I took it out because I didn't, I wanted to leave plenty of time for questions. There's, as you mentioned, there are now sort of these charts or networks, if you will, looking at this. It's a lattice. It's a network of different genetic susceptibility polymorphisms. And there have been both epi studies and also more direct studies looking at GSTP and GSTM1 and besides the glutathione-s-transferases, there are other NQO1 and HO1, and there's a variety of these sort of antioxidant protective enzymes. And I think that there has become, as with most things, we've come to understand this is much more complex than we had hoped, meaning that we had hoped that we had hit on GSTM1 as being, the seminal or the sentinel polymorphism here. But clearly it's not quite that simple, and I guess it's simple-minded to think it would have been. So I agree with what you're saying, and I think that one of the things that we are looking at and others are looking at is what's the panel of genes that would select out this susceptible group. That slide I showed of our group's work showing that susceptibility, where you look at all the different outcomes, inflammatory outcomes, and you can pick out the top 10% that have the most inflammation. If we could take that group and figure out what their genetic background of susceptibility is and then use that to screen populations, that would be the ideal way to do it, because we've taken into account all these potential susceptibility factors rather than one loci. So I do think that's the way things are moving, and of course you can analyze large numbers of genes these days much more cheaply than you could in the past. The issue still becomes in getting it, at least at a local site, and so this is something that might be a multi-site effort. You got to find enough of those subjects to enroll in a study, and so I can tell you, it's not so bad. I mean, GSTM1 is found, I think, 40% of the population, so we didn't have trouble finding enough of those. But some of the other polymorphisms are much less common, and then if you have to find people that have a certain loci at 10 different, or a certain genotype at 10 different loci, now your numbers are starting to get smaller and smaller and smaller. So I agree with what you're saying. I think that's the wave of the future. That's what we're going to need to do to get a handle on this. But I think it's going to take a bigger effort, a more cooperative effort. This is a multi-site type thing to get enough human subjects that you could do the right study and really sort of try to do a definitive study looking at that. But you're right. I absolutely agree with you.
What about you've mentioned a couple of times that the particles were a little different in your exposures. Have you tried to figure out why? Is this an anomalous year? Was the meteorology different? Was there some type of distribution center that was shut down for a period of time? Or, have you tried to figure out what was going on that made your particles have a lower level of PAHs and a lower level of metals?
The short answer is no. We haven't really looked at those factors, and I think that's an important question. We're not aware of any sort of obvious changes in manufacturing or in road traffic or really in anything that would sort of predict this difference. So I think that's a valid question that we need to spend some time thinking about, certainly before we would embark on another one of these studies, because I think that's certainly an issue. We want to be using particles that we expect to have a biological effect.
This is a question from the web. Would be the conclusion to the study that the presence of pH and metals in PM would be much more important for health than PM 2.5 concentration?
Well, I hesitate to draw too many conclusions because, again, we had a small study, but I think that based on previous data and based on what we saw, it appears to me that yes, the metals and the PAHs are likely to be associated with the inflammatory effects, at least in the airway and the measurements that we took. I mean, based on the literature I know, that's been suggested in the past. Animal studies, in vitro studies, it seems to be at least a number of studies, the metals, the the polyaromatic hydrocarbons seem to be linked to oxidative stress and inflammation. And I think that's one of the major issues that we ran into, was we didn't see an effect, and the real big thing that seems to be missing were these particular elements of the particles, you know, with the caveat that this was a small study, and so there's the powering issue also. But I think that's one potential message that could be taken from this, yeah.
Thank you. There's another question. Somebody's asking that she had chronic asthma since years. She was young and tend to be more susceptible during the spring season in California. And you have covered this in your talk, but how was the concentration of various pollens in California measured or not? If not, will future studies consider small to large concentration of common pollens that have a significant impact on asthmatics in combination with other particle types and sizes?
It's a very good point. We did not measure or control for pollens or other environmental allergens in this study. And that's one of the issues that I don't think I said it explicitly, but I tried to highlight in the individual factors that affect human subjects. So the allergic asthmatic who has an intense exposure to an allergen is going to likely that will influence their response or lack of response, perhaps, to the particles. We did try to control for that because patients did spirometry. We had to make sure that their asthma was under control before they underwent an exposure. So I don't think we were seeing patients who were having a clinical exacerbation because of the allergy factor, but at a sort of subclinical level that could affect the measurements that we were taking, which are quite sensitive. That's an excellent point. So I think that going forward, that's another factor that probably should be considered, and controlled for, or at least monitored in some of these studies. And the issue is there are lots of those factors, and you have to kind of try to pick and choose which ones you're going to try to keep track of, because there's a long list of these things that could affect the outcome. But I do think that's an important one.
Thank you. There's 3 questions. One is asking, would there be a way to have measured glutathione levels as an indicator of the patient's reaction to particles?
Yes and no. I mean, there are assays. Yeah, there are assays that can be used, and there are probably people in this room that know how to better answer that than I do. But there are assays that can be used on a research basis to look at that. It's not easy, and it's not been done a lot in human studies. I mean, there are various measures of, quote, oxidative stress, or surrogate measures of oxidative stress, I would call them. So. We chose not to incorporate that. It's something that could be done. I mean, you may know more about those assays. What's that? Yeah, you probably have to do. He said you'd probably have to measure tissue, and so this isn't the question, but to that point, some studies have done bronchial alveolar lavage or bronchial biopsy, which will give you a better tissue measure of what's going on at the tissue level. Again, that does involve risk, and we chose not to go that route. But that is something that more invasively could be done, and might give you a better measure of cellular stress or tissue oxidative stress.
Thank you. There's a second question. Do you think there is just more of a problem of people who are allergic to the particles rather than those who are not allergic to them?
Well, I'm not sure I understand the question. Just to make the distinction, people are not allergic to particles themselves, and that's somewhat important. I mean, when we talk about allergy or allergic reactions, that's when the body makes an antibody, IgE, against usually a protein, but some sort of allergen. And so there's not been any indication that the body becomes allergic or makes antibody to particles, but rather that the particles have a number of compounds that we've talked about, metals, hydrocarbons, and other chemicals that cause inflammation, and may boost the production of antibody to other things. So it's a little bit of a fine distinction, but it's not actually allergy to the particles. But I think the point of the question was, are some people more susceptible to the particle effects and some people less? And I think that's clearly the case based on some of the data that I showed. The challenge that we're trying to get at, and we unfortunately didn't truly accomplish, is to try to figure out who those people are, who's more susceptible and who's less susceptible to the inflammation that's caused by particles.
Thank you. The last question is, do you think the antioxidant enzymes could be supplemented to those who don't have them and used as a therapy for air pollution susceptibility?
If I could have planted that question, I would have. The reason is, so that's actually one of my great areas of interest, is if we think that these enzymes, these antioxidant enzymes are so important, and I think they probably are. I don't think they're the whole story, but I think they're important. Could we increase those enzymes or supplement them somehow, and would that in fact protect people against some of the inflammation that air pollution causes? So that's a personal interest of mine. The answer is maybe. We've actually done, we published a study at UCLA about 1.5 years ago showing that there are ways to boost the expression of these, the GST enzymes at least, and probably some other enzymes, NQO1, HO1, by eating certain foods, believe it or not. And so there's some chemicals in certain vegetables, and we studied broccoli sprouts specifically. But the point is that if you eat enough of these particular foods with these chemicals in it, that you actually increase, and we showed, at least in the sinus cavity, you can increase the levels of the GSTs by 200 to 300% just by eating certain things. Now, the reason I say maybe is that we've not proven yet that will protect you against the air pollution effects. So I can't recommend that as a treatment for anything. But you can upregulate these through certain means. And so we have a great interest in trying to see if that will have protective or beneficial effects. And that's part of the interest in trying to identify susceptible people, because those are the people that we would like to study. We'd like to give these agents to and see if boosting their low antioxidant enzyme levels might actually be helpful to their health.
Oh, just a question. Yeah, quick question. Do you know, has anyone looked at whether or not there are epigenetic changes with either chronic or acute exposure to particles?
Not that I know of, but that there may be some data I'm not aware of. I mean, as you well know, I mean, epigenetics are hot now, meaning that there's a lot of stuff beyond the polymorphisms that is important. And so I must admit I'm not familiar with that data. So if there is something, I apologize. I don't know it, but I've not seen anything that's yet. I know people are talking about it and working on it, but I've not seen anything published. But the truth is, I'm not intimately familiar with the epigenetic data.
Okay, thank you.
Just your previous comment there, about boosting the activity of GST. Obviously, you're not going to do that for GST null or mu1 null individuals.
Right.
They may have some tau allele that could be induced, but not the mu1, you know.
Right, and so...
But there's other systems that could be induced. So I think, you know, if you use an inducer that could induce at many different loci, then that might be effective for antioxidants.
Yeah, and so the agents, the compounds that we've been looking at act through the antioxidant response element. I mean, I showed a slide that looked at that, but the antioxidant response element is responsible for upregulating a whole slew of these GSTs and other enzymes. And so the idea, as you just suggested, was if you're deficient in one or maybe a few that are these that are important at baseline, if you could upregulate the rest of them and sort of cover for that somehow, if you could upregulate this protective network, you may be able to make a difference. You know, and I don't know enough about how high you'd have to go or which other loci you'd have to upregulate, but but yes, we're trying to look at ways to sort of upregulate lots of things from one specific focus, and the ARE is one of those targets through the NRF2 pathway that potentially could be useful, but it hasn't been studied enough to know whether it's therapeutically beneficial. While he's getting the mic, one of the things is that, I mean, as you're well aware, the vitamin supplementation, like vitamin C, vitamin E, has notoriously been disappointing. I mean, there are certain, so that's one of the reasons why we're trying to target things that might have broad effects. Now he'd like to respond, I think.
The mic, just through the mic. Just realize also that vitamin C combined with iron or copper can be a very strong driver of the Fenton reaction because it can cause oxidative damage. Right. So I think one of the complications that we may have in some of the data is that as a matter of fact, I used to use a combination of copper or iron sulfate with vitamin C as my positive control for oxidative stress.
Sure.
Okay, so one of the problems we may have is that some of the procedures for giving these vitamins can be contradictory. Right. Okay. So for example, if you were to give the vitamin without those transition metals, it has a very strong antioxidant effect, whereas you give it with it, it may be actually promoting oxidative stress. Okay, so I think we have to have a little more refined look at some of these issues to really think them through and what might be beneficial or not.
Sure.
I was just wondering about the group, the mild to moderate asthmatics group. So I'm assuming that during the time of the study they weren't taking any kind of medications for their asthma. Were they required to come off of that, or were any people in the group asked if they were taking NSAIDs or other anti-inflammatories, anything like that?
So you're correct. They were not allowed to take any anti-inflammatory medicines, such, inhaled steroids, leukotriene inhibitors, even nasal steroids, anything that we thought might affect inflammatory markers. And they were allowed to take short-acting bronchodilators, though of course there were time period. They had to be off of those, I think, for 24 hours before any procedure. So, but anti-inflammatory medications were an exclusion, and that again speaks to the mild nature, I think, of these asthmatics, because the issue we get into, and it's appropriate, is that you can't enroll a patient that meets the criteria for requiring inhaled corticosteroids and not tell them that you really should be taking an inhaled corticosteroid, but don't do it until you're done with our study. So, we really had to enroll mild asthmatics who didn't need those types of medications, and that's appropriate, but it also, you have to take that into account when we look at the data. Right, the ethics of studying, severe asthmatics and not having their treatment interfere with what you're trying to study is very difficult. Yes.
Thank you, Dr. Ryder, for your nice presentation. With that, I'm going to wrap up the seminar. Thanks everybody for coming. Enjoy your long weekend.
Thank you.